Lynn, I have known for a while. I'm probably best to leave
Leave
give
that
away too
alone.
much about our ages. But I've known Lynn for many years. He's done the most amazing work on Parkinson's. So what we're going to do today, for those who are new to this, is we're going to talk a little bit about Lynn's past, how she ended up working in Pogsies, and then a little bit about how she ended up in the research. And a lot of it's going to be about this research around gait walking and Pogsies, what it means, what the problems are, how can we measure it, and how can we take that sort of information forward to try and help people. As Eli said, we have a Q&A. So if people want to add things in the chat on questions that come to them as we're going through this, do put them in and I will try and pick them up and offer them to Lynn as and when seems appropriate. So Lynn, you're very welcome. You are based in the lovely sunny Newcastle up in the north of England. And it's useful, Lynn, just to hear a bit about your past. So, I mean, what was your sort of background in terms of training, and how did you end up sort of coming to research in Parkinson's?
Okay, well, firstly, thank you for the invitation and to be here today. Really, it is an honor. So it's a long and painful journey. I have to say, probably like many people, I'm fairly old now, respectively compared to many other researchers. But I started out life as a physiotherapist many years ago. It took me a while to start to work in Parkinson's disease,
And
but
was that a
after a few,
physio? Did you train up in Newcastle then? Is that
I
where you?
trained in Newcastle. Yeah, I did my training, and I did my first. I had my first sort of general basic physiotherapy experience in Newcastle, and then I went down south and I worked at Stoke Mandeville Spinal Injuries Unit because I wanted to specialise in neurological conditions, and I was really interested in the opportunity that was available at the time. It was really sort of quite progressive with the neuro rehab, particularly with incomplete spinal lesions. So that
So that
attracted me as
hospital
a
is specialises only in spinal cord injuries. So I mean, basically, it was set up specifically to help people with those type of problems. Is that right, or is it for any
uh,
neurological problem?
both. It's the National Spinal Injury Centre, set up actually after the war because of the number of spinal injuries from as a result of the war. But it also is a general hospital as well. So when I was working there, I continued my general physio experience working in stroke and other conditions. But you also
So
had
can I just
50% of your time with treating people with spinal injuries.
so can I just parade my ignorance here about general physio? So general physio, I always think of physios often as just bashing on people's chest, doing a lot of chest physio for people with chest infections and chronic, lung conditions. And then stroke, it's more about trying to deal with the physio, get their arms working again and getting them walking. Is that what general physio involves? And what's the difference between that and neurophysio, which is where you sort of obviously seem to end up?
Well, physio involves either being in outpatient departments. I think that's probably what most people their experience of physiotherapy actually. So they'll have an ache or a pain and they'll see a musculoskeletal physiotherapist who will provide various treatments to reduce that pain, joint pain or whatever. Um, Respiratory physiotherapy is another specialism, as you've just said. So these would be physiotherapists that would be based not only in intensive care units, but in other departments that are dealing with more chronic respiratory conditions. There are lots of branches of specialty, but I'm describing the main ones. And neurophysio is another area, and that's the area that I was having. I moved through part of your basic training as a physio, and it's the same as a medic. Actually, you tend to circulate through different departments, and it gives you a broad experience across a range of different conditions. So we all did our time in intensive care in the orthopedic wards after people having uh, hip fracture and recovering from knee and joint replacements, those sort of things, and circulating through general medical wards, for example, where you'd be doing a lot of chest physiotherapy as well as general stroke type rehabilitation and
Yeah.
more specialized neuro work. So, and that was the standard way back when I graduated back in the
Okay.
80s. It was the standard to do your general physio training and then specialize after a while, but to get those broad skills.
Okay. And then when you moved to Stoke Mandeville, you'd very clearly decided that sort of neurologically based physiotherapy was what excited you.
Yes, I was a
And
terrible outpatient physio.
okay,
It definitely wasn't the place for me. I was awful with machinery. I shouldn't be admitting to this actually in an
no, because
interview,
when
but
we end up, this will be interesting. Yeah.
it was really not my skill set. So I moved swiftly to an area that I was passionate about, which was really treating neurological conditions and their rehabilitation.
And why
Because I
was
was
that? Why were you passionate about that? What was it that attracted you? Did you feel it was an area that had been neglected, or you felt that was the area you could make the most difference with your physiotherapy?
Um, it, well, in Newcastle actually was a really strong area, and Newcastle is known for neurology. It's often called neurology on Tyne, so it was a really strong area for neurology and neuro rehab in physio. So I had some good role models there anyway from a very early stage. But I
Right.
think it was the for me, it was the my interest in the brain and control of movement, and really trying to then link that to the different conditions that you'd be treating. It was um, and the complexity I really enjoyed. You really are treating the person as a whole. You're not sort of focused in on a particular joint or a very, very specific system. It's a very holistic approach. And I really was fascinated and enjoyed that broader problem solving approach. And that was really very focused on function, actually, very practical things. How are we going to work to get this individual independent again, being able to move around the home, access the community, those sort of things. So I think that really probably spoke quite well to my very pragmatic nature.
Yeah, which is interesting because, of course, we are of a similar vintage in the sense that I think a lot of medicine in the end of the last century was very much drug orientated, and the
Yeah.
idea that there were other specialties that could contribute and help was a sort of it was a bit of adjunct you could do for a bit of amusement, but was it really going to make a difference? And it's clear over the years that it actually makes a huge difference, probably makes more difference than a lot of the drugs we use, especially in Parkinson's. So in terms of your training, you were at Stoke Mandeville, then you went back to Newcastle and sort of started to work within the neuro physio department there.
I moved back up to the northeast for a promotion, and again to the regional spinal injuries unit actually, which was
Mm.
based in rural Northumberland in Hexham. At the time, it's now moved from there, and that was a similar
Hadrian's
sort of
Wall,
setup. Pardon?
isn't it, on Hadrian's Wall?
Near
Hexham.
there, actually, yeah, right out west.
Yeah.
Um, so it was a similar setup, which had a specialized or the regional spinal injuries unit, but also operated as a small general hospital as well. So it was a similar type of experience. I was allowed to continue working in more general rehab and other areas, as well as specializing in treatment of spinal cord injuries, in particular, people that had incomplete spinal cord injuries where there was sparing of movement and um,
Mm.
opportunity really to look at manipulating the nervous system to get the most amount of movement and function. So challenging,
And this
but
would be very
very,
much,
very rewarding.
yeah, very rewarding, but also very service driven. So at some point, you obviously made a decision that whilst providing a wonderful service to help people, you wanted to do some research in it. So when did that happen, and when did you start to transition into Parkinson's?
So my interest in research happened very quickly. Actually, it happened while I was still at Stoke Mandeville, and that's because I was I was very frustrated in being told that I had to just follow certain treatment regimes, which to me looked completely wasn't convinced that they were necessarily the right way to go, and I wanted to know the evidence behind it. So at that time, by the way, there was no research in physiotherapy. It was not. It was this was really way before research careers for physiotherapists. But I had a curiosity about what I was doing and whether it was the most effective way to proceed and if we could do things better, essentially. Um, and when I moved up to Hexham, that desire was still there. And I think it was a recognition as well that personally for me, I wouldn't have really wanted to continue purely as a clinician. I needed to have more. opportunity within my career, and I could really see that coupling that with research, and that ability to inquire and change clinical practice, even though it wasn't called that at the time. But that seemed
Mm.
to me that's something that I would quite enjoy doing that. So an
Yeah.
opportunity came up, as you know how these things happen. An opportunity arose, and I stuck my hand in the air to get involved in some research.
So that must have been quite a, I mean, that was a bit unusual at the time and quite a radical change in direction in some ways. I know it was what you wanted to do, but as you were saying, physiotherapy wasn't regarded as something where research was really a prominent aspect of it. And so you
Yeah.
were trying to move physio into a new area in yourself. And so what was that research project that you did? Was that how you got into Parkinson's, through that research project? No.
No, I've slowly worked my way through muscles and the spinal cord into the brain. It's been a bit of a journey. Um, well, the you're right. I graduated with a diploma. There weren't degrees in physiotherapy at the time, so it was really, doing a
Yeah.
higher degree. Really wasn't anything that people did. And the opportunity was to engage in a PhD, which was rather a large leap from my standard of learning, let's say. But they'd wanted a medic actually, and couldn't recruit a medic. So I said, well, I'll do it. Please recruit me. So anyway, whole story around that. But the research itself was to take an animal model from the cat, around training and engineering muscle tissue. It was a neuroscience, a
Right.
neuroscience type project, and look at manipulating the property of that muscle tissue to see if you could design better muscles. And for those on the call that are of the older generation, in the 70s, the Russians were trying to manipulate muscle tissue to design the best athletes. And we had machines such as slender tone and all of those things to slim people down and improve their muscle tone. So this was of that sort of generation of applying technology to stimulate muscle and to drive recovery. So that was the basis. But this was taking a very specific model from the animal, from the cat, and translating that and applying it in the human, and carrying out experiments in people that had spinal cord injury, in fact.
And you enjoyed that because it obviously didn't put you off research. Because sometimes people say, I'd love to do research. They start research and think, well, this is very tedious and quite hard work. But you obviously enjoyed that, and it
I
sort of
did.
whetted your appetite for more.
I did, although I can tell you it was terribly intimidating. I'd never used a computer at all, and I mean at all. The only thing I knew to do was to take the plug and plug it into the socket on the wall. So let's
Yeah.
say there were numerous vertical learning curves that were ongoing, and I'd never read a research paper. And can you imagine your first engagement with research is to read a detailed experiment
Yeah.
in an animal where they've been, just dissecting it and trying to understand all of that. So there were a lot of new experiences, but yeah, I did enjoy it. I really enjoyed the process of learning. I
Yeah.
enjoyed the process of connecting that learning to, or trying to connect it to my to practice, which was always it's always there for me. I'm always trying to make those connections. How is that going to change practice? Even though it seems very, very remote, you can always make the connections if you're looking for
Yeah.
it. Um,
So
yeah,
you've
and
been
I
very keen
learned.
on the clinical translation and making a difference in people's lives
Yeah.
rather than helping cats. And 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 then after that, so when did you actually start working in Parkinson's? When did you start moving into that field after your PhD? Was it many years, or did you say it was a bit of a long journey and a slow transition, but when did you first start working in that area?
Well, some time later, because after my PhD, I moved overseas for a period of time. I moved to New Zealand and set up a lab in New Zealand and continued my inquiry, working more with people recovering from stroke. And I then moved back to the UK many years later. Um, and when I moved back to the UK, I needed a job. I had bills to pay, and I needed a roof over my head. So there was a research position going in the university, and it was related to developing a new cueing therapy. It was the rescue trial, which is a really old study now, but it was very, very innovative in its time. So they wanted a research um, associate. It was a junior position. Having gone from a very senior position, I went right back down the ladder again to a very junior position. But I thought, well, this looks interesting, and it's new learning again, and here we go.
So that was a queuing device in Parkinson's to help with walking.
Yes, so it's a very simple. Many people with Parkinson's will be familiar with queuing, of course, and well, I think it's probably widely known. Really simple device that um, most typically emits an auditory tone, and that's used to control rhythmic movement, the sort of rhythmic type stepping movement that you would have with walking, and it provides a prompt or a cue that people can use to focus on their stepping and step in time too. So Rescue was an EU funded um, project, a multi-center project with 3 teams based in the Netherlands, Belgium, and in the Northeast that was taking this therapy, developing it, doing some sort of pre-development, and then pulling it through and into a randomized clinical trial.
And it
So it was
worked.
the sort of whole gamut of development.
So it's developing
Um,
the device and then translating it and then doing the trial. And did it work?
yes, we had very good. We had significant findings, but I mean, you know yourself when you carry out research, it's not just what your primary outcome from the research. There was an awful lot of other learning that that happened at the same time. So learning around dual tasking and the role of attention and cognition in movement was a real significant feature, that an aspect of the work that I led on that really made me think more about the control features that are involved in walking in itself. It's not just simply a matter of contracting and relaxing muscles. It's the rhythmic generation, the role of the basal ganglia, but the role of the frontal cortices and other areas in the brain. We had a really novel approach to measuring movement that I loved because I love technology. And this was
Even though you
using a
couldn't switch on a computer.
I know I've changed. Well, I mean, in some things, but not in others. So we had an accelerometer, which was like a telephone directory. This thing was massive, and it was being used to measure movement in the home. Um, and that this was 20 rescue started in 2002, so this was really innovative at the time. I can't claim the credit for introducing the accelerometer, but I can claim the credit for grabbing it with both hands and saying, okay, well, we can use this to analyze gait, to almost have a gait laboratory, and we're going to do it in people's homes. Because all of the work that we delivered in rescue was to run a trial and do all of our assessments in people's homes. We weren't asking them to come in to be assessed. This was a real world study. It was literally the first of its kind, including real world digital measurement. So there was
It was a
learning
problem,
of,
wasn't it? Because basically a lot of this work had to be done in gait laboratories
yeah,
in academic institutions, which are often used for sports medicine and other things. But
yeah,
it was all quite complicated, limited numbers, and as you say, it's very artificial because you're not
yeah.
in your normal environment. So this accelerometer, what was it attached to? Which bit of you?
It was a massive device that had cables coming off it and small accelerometers, essentially, that the wires were connected to a unit that was stuck on your chest and on either thigh. So, and people wore it
Okay.
for I mean, it's a testament to people with Parkinson's. They not only tolerated this, but they wore it for. We asked people to wear it for about 5 or 6 hours of the day, as an evaluation of the effects of therapy. So they wore it when they went about their day-to-day activities at home. I mean, obviously, amongst other outcome measures that we collected,
Yeah.
but that was a particularly novel example of trying to measure real world walking as people went about their day-to-day activities, rather than some sort of weird proxy of that where you walk in a straight line and time it, which is great, it's a good test, but it doesn't really tell you how well someone's functioning. We have to, let's be honest about this. It's not going to tell you if someone can get to the shops or indeed get out of their chair and move to the bathroom and all of those sort of things.
And of course, most people here will know, but in 2002, there was no such thing as a smart watch. I mean, phones might have a camera if you were lucky, but the sort of level of sophistication was very limited, really. So this was a very innovative thing. And then sort of moving on over these years, Lynn, with your research, because obviously this was having grabbed the accelerometer with both hands. You haven't really let go of it over the last 20 odd years, in the sense that you've been very interested in these devices. So it'd be interesting to hear, how have they evolved over the last couple of decades, and what is their value, and what are they like now? Because in my experience, one of the problems with people in gadgetry is they get a bit obsessed with the gadgetry and forget what the question is. And so
Yeah.
there can be sometimes a bit of a disconnect between what's being developed and what you actually want it for. So how has it evolved over the years since that original accelerometer?
Well, the devices are tiny now. We work with a device which is, I haven't actually got one in front of me, and normally I have one hanging around my desk. That's what I'm looking for. But if I'm holding this up, it would be about that size and half
So a
that
couple of
thickness.
so a couple of centimeters by a centimeter by half a centimeter, something like that.
Yeah,
Like a watch, really. Not very different to
yeah, yeah.
my watch. Yeah.
Yeah, exactly. So they're tiny now, and devices now are able to record continuously, sometimes for up to 14 days. They've got multi-sensing capabilities in them, so they're accelerometers. They have temperature and light sensors and gyroscopes, and what that means is that the quality of movement and what you can derive is I mean, it's huge what you can do with this teeny tiny little device.
Yeah.
I make it sound really wonderfully simple. Of course, it's not, but essentially they provide a one-stop shop to measure someone's walking activity, to measure their gait, with the sensitivity of what we can really derive from a very complex laboratory measure. But you can also measure other things. Of course, the signal allows you to infer fatigue. It allows you to have proxy measures of sleep. And you can relate these measures to, you can have more direct measures of function and so on and so forth. So it's from one small low cost device, an awful lot of information can be derived. Or that's the state of play now, and that's where the
So this would be
advances
with a
are now.
wristwatch, or do you still wear it on your chest, or where do you?
Well, we do in Newcastle. We've really focused on a body worn device because we wanted to measure gait, and we wanted to measure things like the movement of how your body moves around, which tells you a lot about the control of your walking as well. And a wrist-worn device gives some really interesting information, but won't necessarily give that level of granular detail into the specifics, the discrete characteristics of walking. So both are good, but they need to be applied to answer to give you very specific information pertaining to what it is you really want to learn.
So you put this device on, and then you collect all this data. So, and what's been the main thrust? Because I mean, as you say, gait problems are well known in Parkinson's. Have problems getting going, gait ignition problems. We have obviously freezing. Sometimes difficulty with turning. I mean, what extra information do you get from these devices, and how have you employed them to understand gait?
Um, well, we used them really to replicate what we could do in the laboratory, but then to understand or describe real world locomotion, because that's entirely different from what someone does in the clinical space of a laboratory. So that really is understanding quite how well someone can navigate and cope with the complexities of the real world. Um, so uh,
So do you
so
concentrate
we
on people? So do you look at people when they're newly diagnosed and say, I'm very interested, and follow them over time? Or do you tend to recruit people to say, well, I've got a problem with walking. Can you help me understand what it is and treat me?
both. So we were interested in, or I was interested in, deploying this sort of technology to address a lot of important questions. So can they be used to help detect Parkinson's
Right.
and complement diagnosis? Can they detect risk of Parkinson's much earlier than before symptoms appear and identify risk? And therefore, if there are new therapies coming through, people can access those therapies at a time when they may benefit most from disease modification, for example. And
So
then
these
can we
are
track?
people with prodromal Parkinson's. These are people who've lost a sense of smell, REM sleep behavioral disorder, all of these type of people, or
Yes.
people from a family, a genetic types of Parkinson's, or
No, we haven't done so much work in genetics, although there are colleagues elsewhere in the world, and at Merriman, for example, who's looked at a lot of different genetic profiles. But we were looking more at people that had an enhanced risk. So we would collaborate with Walter Maitzler and Daniela Berg, for example, who had a risk cohort. And
in
Walter
Germany.
approached
Yeah.
us. Yeah, that's
Yeah.
right. Walter approached us to use the skills and the knowledge that we developed in the laboratory in gait analysis, and we'd really translated to using these devices and sensors. They'd collected data with sensors, but they didn't have the analytical skills and knowledge that we had. So we brought these aspects together, and actually it's Sylvia Deldin, who's in my group but has her own research group now, did the analytics because she was really working with me with digital devices at the time, advancing how we measured gait, and then what we could use those measures to tell us. Really, it's not just, oh, that's an interesting measure of gait, but can it help us with diagnosis? Can it help us identify people early? Can we track change over time in a way that's more sensitive? Can we evaluate the effects of intervention in a more sensitive way as well? So they would be examples of how we would employ these outcome measures or these devices to have more enhanced outcomes that would not only be useful in physiotherapy. Obviously, I'm interested from a physiotherapy perspective, but they would really provide complementary tools in clinical care, but also in research that really still are urgently needed.
Okay. I'm just going to go side tangent because one of the questions here is about the availability of these devices, and I guess there's two questions I'd have with that. One is, are the devices which you're using widely available? So are they available in the US? And I guess listening to what you say, part of the problem is not so much having access to the device, but having access to the analysis of the data that comes out of the device. So you
Yeah.
can collect all this data, but if you don't have the expertise which you clearly have developed for analyzing it, it provides some information, but not a lot of useful information. So how widely available are they, and how widely available is the analytical pipeline that goes with you to enable you to actually understand what data is coming out?
So everyone is familiar with wearables. I mean, most people have, your phone is essentially a mobile device,
Mm.
and we're all wearing them now. So there's a whole raft of commercial devices available. We don't use those commercial devices. We use research type devices that can be bought and can be made available anywhere. There are really good systems, for example, the from Clario Precision Motion, that measure people's movement as they go around. That was developed in the States by physiotherapists, actually, Faye Horak, who really stimulated that development. But there are other devices. We use an activity device here, and there are Mac Roberts devices. There are others that can be used that give you a more sensitive signal, let's say. But you mentioned this earlier. People get a bit confused about devices versus the algorithm, the mathematics that you use to apply to the signal that the device generates from the sensors within it. And it's that combination that really tell you something interesting about what you want to measure. So we are more interested in, or have always been, device agnostic. By that we mean that I mean, and this is, by the way, the direction of travel for the in research. It's not the device that's important as long as it has the right technical characteristics. It measures data at the right frequency, those sort of things. You wear it in the same place, you have a similar protocol. It's the algorithm that you apply in a standard way to the device that you use in a standard way that generates the information that you need. So that would be called robust technical validation. And we
Yeah.
also need to know that patients are prepared to wear it as well. Otherwise, it's all hopeless if they go, no, that's rubbish. I'm not prepared to engage with you. And if clinicians and researchers are prepared to use it. So I think I'm going off tangent a bit, Roger, so pull me
No,
back.
no,
But
it's important to
essentially,
understand how these devices, because I think there is a sort of
yeah,
perception, if I get my device, and then that's it, I've got everything I need. But actually, what you're saying is that you've got to make sure you're getting the right information, then you analyze the right information, you analyze it in the right way to get what you need.
that's
And I
right.
suppose coming back to your original question you were asking, did you pick up abnormalities? Because presumably these are people with prodromal Parkinson's, REM sleep behavior disorder, and such like, who aren't aware that they've got any walking problems. Did you pick up subtle abnormalities on those? And then how did they track across disease? Did they get worse as people progress, or did different people follow different courses?
So we did. Um, and we detected, because these devices are measuring at 100 hertz a second, so you can detect really, really sensitive changes. 100 hertz rather, and you can detect really sensitive changes. And what we found, I think it was about 4.5 years prior to people converting, this is in a cohort of over 600 people who had a higher risk of developing Parkinson's. They were followed up, Daniela and Walter's cohort, and they were followed up longitudinally and until the point of developing Parkinson's. So 4.5 years prior to that, we were able to detect differences in the variability. So how variable one step is compared to the other. So the variability of timing of movement as well as asymmetry of movement. So these are things that you wouldn't necessarily be aware of because they're really happening at a level of minutiae, millisecond level that none of us
Yeah.
would be familiar with. So that they were able to predict conversion to Parkinson's disease. However, when we looked at what tracked in the intervening period up to that point of diagnosis, unsurprisingly, it was things like walking speed and stride length that then started to decline. So what you can use as a measure that will identify risk will be different from a measure that will identify progression. And that's quite important as well when we start to think about using these devices. Often people just think, oh, well, I just need one measure from it and that will serve all
Yeah.
purposes. But that's not the case. These measures really can relate to different areas of the brain that are involved in locomotion, and even the asymmetrical aspect. If you think about Parkinson's being an asymmetrical disease, or condition rather, then we would see more asymmetry. And actually that translates to less asymmetry over time because of course both sides of the brain catch up. So it's really complicated how you actually interpret the outcomes and the data, and relate and correlate them with the underlying control of movement, but also the underlying condition itself.
So that's very interesting. So basically, there is a set of parameters which allow you to be more confident about a diagnosis. There's a different set of parameters which measure disease progression, if you like, and that is a dynamic process. So that what tracks a certain part of the condition at a certain stage may change at a later stage. So one could imagine, for example, it's asymmetry, then it becomes stride length, then it maybe falls or something like that. So you can imagine your outcome and what you're measuring may be different. And if you ask these people, you say, actually, we've got this data on you. There are a few changes here in terms of which we're picking up. Are you aware that there's anything wrong with your walking? Were people aware of it at all, or was it actually, when you say, well, you're slowing up, your stride length's getting smaller, a bit of asymmetry here, a bit slower on that?
Well, unfortunately, they weren't interviewed 4.5 years earlier, and that would be really interesting to ask people. Um, I mean, we can sort of try and, well, I use the word hypothesize. We could try and hypothesize what that might feel like.
Yeah.
And my hunch is it would feel like you just are less smooth and confident. It's just not
Yeah.
as easy as it was. I don't think people would really necessarily notice slowing at that stage or even
Yeah.
reduce stride length. I mean, these are the sort of classic signs that you would see much closer to
Mm.
diagnosis. But I think there would be, it's the fluent, really those measures are talking about the fluency of movement.
Yeah.
And it's such a subtle, it's a real qualitative thing, isn't it? It's the
And
quality.
it's, yeah,
Um,
it's also quite interesting, I think, because sometimes, I mean, certainly with dyskinesias, which I know are slightly different, individuals with dyskinesias might not even recognize they've got them, whereas the family definitely recognize it. And often in clinic, when I'm diagnosing in the early stages, they're not aware of the fact that everyone's having to wait a little bit for them to catch up on the
yeah,
walking. It's other people that notice it more than them.
yeah,
Um,
yeah, exactly. So yeah, that having that external reflection really.
yeah.
Um,
And how do you disentangle? So one of the questions we've got is obviously there are questions about diagnosing early. If we haven't got therapies, what's the point of that? But at some point we have to diagnose in order to help us with the diagnosis and give us greater confidence. But secondly, when these therapies do come in clinic, we've got a cohort that are ready. But the question I wanted to ask was, how do you factor in other problems? So of course I might end up with a bit of asymmetry and a small stride length because I've got a bit of a gammy knee or a bit of an arthritic hip, or I've got a bit depressed or I'm a bit tired. So how do you disentangle getting older and mechanical and other problems with your walking in your models?
Very good question. Well, you have a control population who are older that have all of those conditions as well. So we've always had control populations and have asked the question, what is changing in a population of the same age and same characteristics that's different in Parkinson's? So what's related to Parkinson's and what's related to aging? And that's really, really important to then be able to separate those things out. Clinically, of course, when you sat in clinic and someone comes in, that's just clinical. That's a good clinical exam, isn't it? And an
Yeah,
interview asking about general problems and aches and pains and figuring out what's arthritis, which we all have after a
yeah,
certain point, and what's Parkinson's. So
yeah,
really trying to disentangle those things because the treatment is different.
yeah,
So you would formulate a plan that was very different depending on what the answer was. So that's for me is that's just good clinical practice and clinical reasoning, as opposed to your other question, which is, okay, well, you say you've got this fantastic measure and we're going to use it to identify prodromal Parkinson's or progression over time. Well, it better make sure it's actually looking at progression in Parkinson's rather than progression in your arthritis. They're
yeah,
both very, very important, but they're tackled in a slightly different way. I mean, ultimately, if you think about these real world measures, when we
yeah,
get, in a few years' time, these will be the truly personalized measures, won't they, of your mobility as you go about your day-to-day activity. That's going to account for some of those age-related things as well as the condition itself. And broad models are models of a population as opposed to individual models of the person. So we're going to have to get to a stage where we're trying to balance those two interests. We're not
yeah.
there
And I
yet.
want to come back to that, really, personalised
Uh-huh.
versus population, because I think it is very interesting what you're saying. Because you certainly, sometimes when we're doing studies and people come along and audit in, they say, well, how can you say this person hasn't got X when their walking is so bad? And you say, well, their walking is so bad because they've got two completely stuffed knees and they can scarcely walk. So the reason they're unable to heel toe or they don't have any balance is not related to their neurological problem. It's related
Yeah.
to their orthopedic. But the question I have is, and you seem to, you've already really sort of answered this, that I mean, obviously, you can take populations of patients and you can say, well, I've got 100 people with Parkinson's age 60 to 70, and I've got 100 age and sex match controls, and there are differences between those two. And therefore, in Parkinson's, we can see X and Y. But what you're saying is, I can take Mr. X or Mrs. Y out of this 100, and I can look at them on a personal level and say, I can see your walking's changed. I can track you as an individual and I can tell you why your walking is a problem, which is different from Mr. Z or Mrs. A or something. So you can do it on an individual basis, and then presumably the treatment could then be very much geared towards an individual person rather than all 100 of you are going to get this form of therapy to try and help.
Yeah. And people with Parkinson's are not interested in the 100. They're quite rightly interested in themselves, as I
Yeah,
would be, as we all are. Um, and when we're talking about the use of these tools, every time actually the last World Parkinson's I presented on this topic, and course, the audience say, well, that's all very well, but when am I going to have these tools to use? Because they're really useful to help manage your own mobility and your own condition. To give you an insight into, well, how active are you? Not just how many steps you take. That's a pretty blunt measure, actually.
Yeah.
Probably what's more important is how many times are you getting up and moving? And when you are moving, can you stay on your feet for a reasonable period of time in order to carry out a range of functional activities? These are measures that we would call not just the amount of movement, but the pattern, someone's pattern of movement as well, because you can start to infer more about their function. And the patterns of movement are often very sensitive to Parkinson's. We know that people lose the ability to stay on the feet and stay moving. So thinking about that clinically, if I was having a conversation with a patient, we would be very much talking about, okay, well, this is what we're going to work on. We're going to work on your endurance to stay on your feet for a bit longer and your confidence to do that. I didn't have access to these devices when I was in the clinic. We've really, I should talk
Yeah.
about mobilized in a second, but they're really now the accuracy of the measure is such now that we should be able to start to at least have those conversations. But you then are able to say, well, we're going to try and change this. And that would be the thing that I would want to monitor with my patients. I used to monitor it through diaries and ringing them up to say, all right, then are you getting out?
Yeah.
Are you out and about? Are you walking around the block? Are you able to do this? So it was a more crude version, but essentially you're doing the same thing, but you're just instrumenting it and giving the tools into the hands of the individuals that need them.
Yeah, and I guess there's always a tension between that, because as you know, you can sort of say, well, it's terrific. Your stride length has increased by 1.5 centimeters. This is a huge success. And they'll say, yeah, but I still can't get to the shops or go around the
Yeah.
garden or whatever. So you have to sort of measure the objective measures versus the real world. What difference does it make in my life and where I can move around? So at the moment, what's your main area, Lynn, in terms of how you're using these devices to inform therapies? And one of the questions which has come up is around, should this be the direction of travel for disease modifying therapies? I mean, based on what you've said, you can pick it up. So why do we still use the UPDRS? Why do we still use these measures, which are objective because they give you a number, but they're very subjective depending how the person's feeling that day, both the examiner and the person with Parkinson's? So why aren't we using these measures, and what are you doing now with this information? What's the current direction of research for you?
Well, that's a fantastic question, because that basically is what is engaging me completely at work now. There is a huge drive to use these measures, particularly these real world measures, because they give meaning. They're measuring something that is meaningful to individuals' mobility, as opposed to finger tapping and aspects of the UPDRS, which is much more abstract and arguably doesn't really matter to individuals. So there's a huge drive to develop these measures for use in research. The pharmaceutical industry, of course, are extremely interested in having better objective measures, but also the pull through into clinical care as well. So there are a number of large initiatives underway and have completed, and our work is continuing. I was fortunate to be the coordinator of something called the Mobilise Consortium, which was an EU-funded consortium. It brings together public industry, pharmaceutical industry, and technical companies together with an academic consortium. And our remit was to develop a measure, digital mobility outcome measure, that could be used not only in Parkinson's, actually, that could be used generically across a number of conditions, and ultimately would be adopted by regulators so that drug companies could use these in drug discovery, and physios would use them in rehabilitation studies, and ultimately they would start to inform clinical decision making as well. So,
But I mean, regulators are, well, not a regulator obviously, but they always seem a little bit reluctant to adopt these so-called surrogate markers. There seems to be a big push with regulators for quality of life and patient-related outcomes, which is understandable. And yours almost go in the opposite direction. They're not patient-related outcomes; they're sort of digital outcomes. So how has been your conversation with the regulators? Have they been rather receptive to this idea that you can use these measures? And I guess coming back to a question or we discussed earlier, I guess your digital measures must in some way map onto patient-related experiences. Otherwise, they're not necessarily that useful. So how much have you moved the regulators? Do you think they are receptive to this?
Um, I think within Mobilized, it finished last year and is continuing with another funding stream. We made significant strides. The regulators are very,
Very
very
good,
interested.
significant strides.
So the regulators are really interested in these novel digital endpoints to use their terminology. Um, but it's a very conservative and slow process to get
Yeah.
there. So we made we did make significant advances in Mobilize. We had rounds of qualification advice where you took what you were doing, all of our research methods, to the regulators and said, this is what we're going to do in developing this new measure. Do you approve of the way we're going to generate the evidence? And we've had our methods endorsed. So that means it gives you greater likelihood of success. We were able to very accurately measure a whole raft of different digital mobility outcome measures in Mobilized, thankfully, and show that they're clinically valid. But some gaps remain in our knowledge, and those gaps are in their use in clinical trials, where they would be used as ECO as clinical outcome assessments rather than patient-reported outcomes. So they would give objective measures, but importantly, the measures are objective and meaningful in that you're actually measuring someone's walking. You're actually measuring a meaningful aspect of health. And we've carried out qualitative work to endorse that. And of course, we know that regulators want to, if you're testing a new therapy, you need to demonstrate that it changes how someone feels, functions, and survives. And these are tools that directly speak to the need to generate evidence that will identify how someone is feeling, their quality of life, how they're functioning, and actually we're looking at prediction of survival as well with these measures. So we're still mining the data. We did a big clinical study and mobilized in 2,500 participants, collected data every 6 months to show A, we could deploy the methods, but B, we can then start to generate this additional piece of evidence. So there's a huge interest. But there's still some gaps remain. I mean, I would say that and quite right, in disease modifying therapies, these are the sort of tools that you want to use to be able to deploy in real world environments as well, to have more inclusive clinical trials, include the right populations, deploy them in the real world, but have more sensitive measures that are going to detect a signal of change, hopefully earlier than the clinical standards. And we all know the limitation of those clinical standards. They're just simply not sensitive enough. They're not good enough for the job that they need to do. So
and so looking to this sort of next stage then so through the work in Mobilize, that would be, we would now have measures we could start to incorporate in clinical trials. So if I was to come to you and say, look, I want to start a clinical trial tomorrow in people who are on the cusp of getting Parkinson's, I want to run it over 3 years, I've got this drug which I think might be helpful, you would say, we can help you with these digital measures, we can analyze them for you, and that at the end of that, if we see a change in those, that we know that they will be clinical relevant because they're measuring something which we know is clinically relevant to the patient, and the patients would agree with that rather than we just changed a parameter. And linked to that would then be, obviously it's accessibility. So we can post the devices out to everybody, but is the analysis all still being done in Newcastle or wherever? And actually, that's the bottleneck. I can't take a device here, plug it into my laptop here, get the data, and say, right, I know what's happening to my patient.
So Well, very excitingly, I don't know, you'll know of Camille Carroll and Tom Fulton's EGS ACT Consortium, which is a multi-arm, multi-platform trial, one of its kind in Parkinson's disease that's looking at multiple disease-modifying therapies. And the trial design has a faster throughput to generate evidence. Embedded within the EGS ACT consortium is a funded digital sub-study that's going to use the mobilized methodology to collect digital outcome measures in every participant that's going through the EGS ACT trial. So we're carrying out, and that's the largest interventional study. We have deployed our measures in a range of different clinical trials, actually, but this is a first, and that's been led by Alison Yarnall in Newcastle, supported by the team here as well. So that's going to generate, significant game-changing evidence. It's really exciting, and it's tied into disease-modifying therapies, which, of course, is a pipeline of therapies available now. But the question as to, are you doing it all in Newcastle? And is that the bottleneck is a really important one. We took steps in Mobilize to try and get around that question and to try and start to move these tools into more widespread use. So the algorithm that we developed in Mobilize that generates these measures, we released openly for research and commercial development. And it's difficult. You can imagine from a university perspective who want to spin out IP, but myself and others in the executive were really determined that this was going to be something that we gave back, because actually all that's going to do is get people to use it and improve things. The pipeline of analysis is challenging. But we know that 4 companies have already adopted the mobilized methodologies, and they have their own analytical pipelines as well. So you could go to a commercial company now and say, I want these algorithms, I want these measures done in this way, and they will produce those outcomes for you. We have our own pipeline in Newcastle as well. That is, we've developed over many, many years, which fed into what we did ultimately in Mobilized in generating an analytical pipeline. And yes, we use that, but we actually work broadly with lots and lots of different people and are able to really run a fairly efficient system now, because we're used to all aspects of acquiring the data, cleaning the data, QCing the data. We have really fantastic staff who will train people across multiple sites. So there are multiple ways to tackle this, but that's the way that we're taking things forward in Newcastle, while also releasing work and knowledge and training more widely so that other people can pick this up and adopt it. But the reality is, like imaging data, it's really difficult data to work with. It's not simple. And you wouldn't just chuck an image at someone and expect them to know what they were doing. We have to, people have to realize it's really important to understand that this isn't just something that, your watch on your wrist says you've got X number of steps. It's a much more complex process, because ultimately the measures that we've made some significant progress in are going to be the ones that tell people that the therapy is working, and they're going to be robust, and they have to be robust. So still some work to go, but I'm very confident that we are making progress, and it's not an if, it's really a when, and that when is in a very relatively short duration would be my summary.
So we're running out of time then, but I have 2 questions for you. So one relates to what you've just said. So obviously this is the World Parkinson Coalition. So you talked about the UK study, this multi-arm, multi-stage study, the MAM study. Am I right in thinking that your aspiration, and actually the direction is that if I was in Australia, New Zealand, America, India, all of this would be accessible to me, both in terms of device and analysis. So this really could become something that could stretch across the globe, so everyone with Parkinson's could benefit from this. I mean, from what you're saying, that is what I've understood it to be.
Absolutely, it should be the global standard. And and as an example, we carry out research in sub-Saharan Africa with Professor Richard Walker, so where we've got experience of deploying these tools, and we work with colleagues in the States and in Australia and New Zealand. So these are generic global tools. They will be used generically in a global way eventually.
Okay. And so my final question before we wrap up is, we've talked a lot about drug therapies and everything else, but presumably, these devices are used to record things about gait, and the physical therapy where you began could be interventions that we should be thinking more about.
Well, precisely, because of course it's not just taking a tablet or having surgery. Movement and exercise is really fundamentally important for all of us, and these devices will measure the effects of a wide range of therapies that and activities that people should be engaged in.
Yeah. Well, I think that's a great thing. Having just taken up Pilates myself, I didn't realize just how inflexible I am until I was demonstrated, and how useful these physical things are for keeping you mobile and such like. So we're sort of running out of time. So I just want to thank you, Lynn, for fantastic hour of discussion. I mean, I admire you enormously for the work you do. Say the fact that you've come from an area which was not traditionally academically minded, and you forged a route which has been fantastic. And it hasn't just been academic research for academic research. You've always kept a very clear focus on helping people with Parkinson's and helping the population. And your work has been outstanding in that, collecting information in a much neglected area, which we can use not only to treat populations, but individuals. So I want to thank you for sharing your story with us and all the work you do, and the fact that you're very cognizant of the fact you want it to go to the world Parkinson community and everyone to benefit from it. And in that respect, I just want to remind people about the WPC, which will be next year in Phoenix, in Arizona, in May 24 to 27. Registration's open, book for it. It will be a fantastic meeting. We will hear more about walking and gait and digitals and all of that. So thank you very much, Lynn. Thank you very much to the audience today. You've been terrifically interactive. I very much enjoyed all the questions, which have been absolutely fantastic. I would like to thank Supernurse Pharmaceuticals for funding our research spotlights. And I'd like to remind everyone that the next one of these will be on Tuesday, December the 2nd at the same time, which will have David Stannett and Karen Jora-Ray, who works with him. They'll be discussing their research in Parkinson's disease. So thank you all very much for attending today. Thank you again, Lynn, for a fantastic hour of discussion. And I look forward to seeing you on December the 2nd, but more importantly, May next year in Phoenix, Arizona. So goodbye, everyone.